Hi, Gen Bio. Dr. Fry here. We're back for spring semester. Yay. But unfortunately, we won't be able to see each other for the first 2 weeks, as I'm sure you know. So for the first couple weeks, we're going to have extra videos for you guys to look at. We would normally be doing dissections the first 2 weeks, which I'm so upset that you won't get to see in person. But we do have videos of those. And when you do return to campus, we will offer you the option to come and look at these dissections and perhaps do them in person so that you can see the real live specimens. It is regretful that I am regretful that you won't be able to do those in person with me this week. So we're going to talk you through the lab, and we have lots of resources for you to look at. And maybe when you do get back, hopefully you'll have a chance to come and look at the specimens in the lab outside of our normally scheduled lab time. Okay. So first, since this is our first lab, I'll just introduce myself. If you didn't have me last semester, I'm Dr. Colleen Fry. My email is the best way to reach me. It's cfry@lvc.edu. And the lab will take place on Tuesdays in Knighted Garber 317 once we're back. You'll be put, same as last semester, into a group that will either be from 8 to 10 or 10 to 12. Okay. So just a few logistic issues first. Make sure you're on the Canvas page frequently. As you know, we're going to be relying on it heavily to present the material ahead of time, just like last semester, and add additional resources. Okay. If you can't get on that page, shoot me an email. We need to make sure that you are on there frequently. It's where we're going to post everything you need. Okay. And then the syllabus is already up there as well. Make sure you read it, print it out, highlight the important dates where things are due. I'm not going to be after you guys to submit them. You guys are in college. You got to look at the syllabus for the due dates. Okay. Don't forget that you have an assignment due. Okay. So just to give a brief overview, we have different writing assignments for this semester, which will teach you how to write a scientific journal article. So you're probably, I hope, familiar at this point with the basic parts of a journal article. When you're writing for science literature, you need to include an abstract, an introduction, materials and methods, results, and a discussion or conclusion with graphs as a part of the results section. So all of that compiled will be a complete journal article. And throughout the course of the semester, we want to walk you through each of the parts, how to format them correctly, and really make that a strong asset of yours to be able to write for science literature. Okay. So we're going to break those assignments down into different sections first, and then have you write a complete lab report by the end of the semester. Okay. So the first two assignments that you will do will introduce you to plagiarism and writing citations appropriately. So as far as citations, we're doing an in-text and an end-text citation. So the first one would obviously be like if you were citing something in your paper and you had to reference it, you'd say in parentheses, Fry, and then the year that I published the paper, say 2014, whatever it was, Fry 2014. And then you'd go to the end citation, and that would give you all the further information that you need. So you'll be responsible for writing those the first week. And then throughout the semester, we do the other writing assignments and then conclude with a full lab report, which would basically be a journal article. Okay. The equivalent of that. And then we have some McGraw-Hill assignments this semester, not for every lab. I believe it's 5 total labs. They're 2 points each. And then for lab work, after the lab, we again have 5 different assignments that you will do, 2 points each for some of the labs that will make up the assignment portion of the lab. And then you also have 3 exams. Those are covering, the first one will cover labs 1 through 3, the second will cover labs 4 through 7, and the last exam will cover labs 8 through 10. And then, similar to last semester, you won't be tested on the last exam. The last lab exam will not have material on it, questions on it from our last lab, but we will have an assignment for that, which you will earn points for. Okay, so that'll be separate from lab exam 3. Okay. And then, same as last semester, we combine those points with your lecture for the one total grade. Okay. All right, so now let's look at our material for week 1. We're going to move to talking about protostomes. Okay. And the crayfish and the Nereis worm are going to be our model organisms for protostomes. Okay. And before I get too far into what protostomes are and the characteristics associated with them, I'm going to make sure that you know your directional terms. When we're talking about anatomy, and if you're looking at the dissections, you definitely need to know a few terms. Okay, so if you don't know them already, rostral is towards the nose. Okay, towards the head, towards the nose, that direction. Caudal is the reverse, towards the tail. Okay, dorsal is always going to be towards the back, and that doesn't matter if we're talking about a 4-legged or 2-legged organism, it's towards the back. Ventral is towards the chest. Okay, it's not oriented in space, it's oriented with regards to the body. Okay, dorsal back, ventral chest. And then median is, of course, the midline. Lateral is going to be towards the sides. And then proximal is just closer to, and distal is further from the body or from the midline. Okay. All right, so you should know those. Let's move to our phylogenetic tree. So the first 2 weeks of lab, and I know in lecture as well, you're talking about different classifications of animals. So you should be familiar with a phylogenetic tree of the animal kingdom. Okay, so that's what we're showing here. This is a phylogenetic tree with different points of divergence, different branches on it that separate out the organisms based on similarities or differences. And these similarities could be morphologic, meaning how they appear. They could be genetic, so we could compare the gene sequences of 2 similar organisms, or they could be developmental comparisons. So even if an organism has a similar structure to another organism, it might be formed in a different fashion. And so these characteristics are going to help evolutionary scientists classify animals as related, as more or less related to one another. Okay. So there are 4 major points of divergence on this particular phylogenetic tree. Okay, so I'm going to talk you through those. The last one, we're going to look at the next slide just for a little bit more detail. But the first one, well, we're starting with metazoa. Metazoa are all animals, okay? The first point of divergence for the animal kingdom is separating out what we call eumetazoans, true animals, from those that are not really true animals. All right, they're parazoa. And we distinguish these 2 based on the fact that eumetazoa have what we call true tissues. So specialized cells working together for a common function. Tissue makes up a tissue. If you remember from last semester, we looked at muscle tissue, nervous tissue, and so forth and so on. Versus, say, a sponge here in our parazoa group doesn't really have a nervous system structure. It doesn't have muscles. It's kind of just sitting there and eating. So there's not, the sponges don't have full tissues the way that all the other animals do. Okay. So that's the first major point of divergence in the animal kingdom. Okay. And then the second point of divergence separates animals that have bilateral symmetry, the bilateria, from those that have radial symmetry. Okay, so radiata. Bilaterally symmetric animals, you can cut down the midline and they're symmetric left to right, fold it in half like a piece of paper, right? Radially symmetric organisms, however, like jellyfish you can cut on any plane and fold it in half and it's identical, right? It's round, it's like a circle. So associated with symmetry often goes whether or not the organism has 2 or 3 germ layers. Okay, so bilaterally symmetric animals will have 3 germ layers: the endoderm, the mesoderm, and the ectoderm. Okay. Radially symmetric organisms only have 2. They are diploblastic, 2 germ layers, and they just have the endoderm and the ectoderm. Okay. And then the third point of divergence is the presence or absence of a body cavity. Okay. So this body cavity is called a coelom. It's a space, it can be fluid filled space that separates out basically the digestive tract from the outer body wall. So you think of your body plan as a straw inside a pipe. So there's 2 cylinders, right? One big one, one little one. You've got a tube, the straw, mouth to anus, which forms your digestive tract. And then outside of that, you got kind of another tube, which forms the external parts of your body and includes your muscles, lots of nervous system structures, skin, all of that will be the outside cylinder. Okay. Now between these 2 cylinders, there's a space, and it can have fluid or gases, etc. That's called the coelom. Okay. So whether or not the animals have body cavities will be another point of divergence in the animal kingdom. Okay. And then we get to our fourth point of divergence, which separates our protostomes from our deuterostomes. And these, I'll remind you, are the 2 different groups we're going to look at in our first week of lab and our second week of lab. Okay, so we're looking at protostomes this week and deuterostomes next week. Okay, and this divergence is based on how the coelom forms. Okay, so that space, how it forms, is different developmentally in the protostome group clade and the deuterostome clade. So in order to look at that more clearly, let's move to our next slide. Okay. So you might remember from last semester, hopefully, that we looked at the early stages of development. I think it was in C star. We had 1 cell split into 2. We had 2 cells split into 4. We went through cleavage, and we got to the point where the organisms were what we called blastulas. Okay, so a hollow ball of cells right here. Okay, and this is the same for protostomes and deuterostomes. However, there is a point shortly thereafter where a difference in development takes place. And this is where we separate out our protostomes from our deuterostomes. Okay, so you probably remember that after you had your blastula, there was a little dimple up, a little divot that started to form in the blastula. Okay, and that was called the blastopore, first indent into that ball of cells, right? Okay, so that's again the same for protostomes and deuterostomes. It will lead to the archenteron. Hopefully you remember that term as well. But the mesoderm tissue that starts to form is different in protostomes and deuterostomes. So the mesoderm is going to be these red cells that you see in the picture here. At the top, you're starting to see the mesoderm cells form and move starting from the entry point of that blastopore. Okay, so this red cell structure is going to divide. These cells are going to migrate, and they're going to form a space between your digestive tract and the outside of the body. Okay, so they start down here where the blastopore first appeared in protostomes. In deuterostomes, the mesoderm tissue forms like little buds, little outpocketings from the archenteron side. And then those cells are going to move towards the blastopore. You're going to have division and migration of these mesodermal cells in sort of the opposite direction. Okay. it's going to result in the similar structure where you've got the mesoderm layer surrounding the external side of the digestive tract and the internal side of the body wall. And you're going to have that space in both called the coelom, okay? But the developmental process is different. And so that's how we classify and differentiate protostomes from deuterostomes. One other important thing I'll mention in the development of protostomes versus deuterostomes is that the blastopore, the fate of the blastopore, is different between the two. So in protostomes, the blastopore becomes the mouth. In deuterostomes, the blastopore becomes the anus. So the tube is still formed, but it's almost backwards from the other group. Okay, so in fact, I'll mention that protostome, it will help you remember if you can break down that word. Proto means first, and stoma means mouth, I believe, in Latin. So first mouth. So that first indent, the blastopore, the first indent into that ball of cells is going to be the mouth, protostome. Versus in deuterostomes, second, deutero means second, stome. So the second pore is the mouth. The first one is the anus. Okay. All right. I hope that's clear. Let's move on and look at our model organisms. Okay. So the two that we're going to look at are the crayfish and the Nereis. Okay, they're both protostomes, and so they both exhibit bilateral symmetry, and they both have the blastopore forming the mouth, and then they also exhibit what we call segmentation very clearly, especially on our Nereis. Each of these segments is called a metamer. And most of the segments have appendages associated with them. You can see the little legs. These are called parapodia on the Nereis worm. So there's appendages on almost all of the body segments or metamers. For the crayfish, there are body segments as well, although in some places it's not as visible. Okay, so in the cephalothorax, you don't see those metamers as clearly, but on the abdomen, you absolutely can. See, here's one, a second, a third, a fourth, and so forth. So they both exhibit segmentation of the body. Again, most of the segments will have appendages associated with them. It's when we look at the ventral side of the crayfish, this is a picture of the dorsal side. When we look at the ventral side of the crayfish, you'll probably be able to see that a little bit more easily because of the attachment point of the appendages associated with the different body segments for the crayfish. Okay, so let's look at our Nereis first because I feel like it's the more simplistic of the two. Okay, so it's in the phylum Annelid, which means ring. You can see that each of the repeating rings separates out a body segment, a metamer. Okay, and it's in the class Polycate, which means many bristles. As you look at the parapodia, you'll understand why it takes that name. And then the genus is Nereis. Okay. So the next slide shows some external as well as internal anatomy, which you will be responsible for. Okay, the leftmost picture is looking at the ventral surface of our Nereis, so the chest. This top right picture is showing you the head region of the dorsal surface, okay, of the Nereis. And then, of course, we're looking at some internal anatomy in our right-hand picture here. Okay. So I'll first just mention that you can see all these metamers, all these body segments on the outside, and they each have little legs associated with them. These are parapodia. Okay. Well, I shouldn't say they each because there's at least one that doesn't have parapodia. So this segment here, called the peristomium, is the segment that is just anterior to the first metamer with parapodia. Okay, so it's just in front of the first segment with legs. Okay, that's called the peristomium. And then there's a segment just in front of that I'll just draw your attention to this picture here. This is the peristomium from the dorsal side. And then just in front of that is the prostomium. That's the segment that's just in front of that peristomium. And it has what's called palps. This is one palp. This is another palp, little protrusions on there. Okay. And then coming out from the junction between the peristomium and the prostomium are what we call peristomial cirri or peristomial tentacles. Okay, all of these little appendages up towards the head, those are tentacles. Okay, peristomial and then prostomial tentacles. Okay, and if you look closely, you can see that there are little eyes on the prostomium as well. And then there are some mouth parts, which I'm going to talk about in more detail in our next slide, okay? But I'll just mention that there are some mouth parts which can change, okay, based on whether or not the organism is feeding or not. It has the ability to evert its pharynx outside its body to grasp food. So you'll see these jaws here, these little sharp pinchers are actually not always outside the body. They're extended when feeding, okay? You can see those here as well. So in this picture, we've got what we call an everted pharynx, okay? So the pharynx is outside the body, allowing those jaws to be outside and to grasp and pull food in for digestion, okay? All right, so this is, I think we've covered most of the external anatomy of the Nereis. So let's look at our internal anatomy. If you were to flip this guy onto his belly, right now we're looking at the belly, the ventral side, flip onto his belly and look at the dorsal side. That's what you'd be seeing here. You would cut the body wall with a pair of scissors, sort of in a cross section type fashion, and then cut up through the back of our Nereis worm and try to, we always advise you if you're doing this in lab to try to avoid cutting on the midline because you don't want to cut the intestine, which is what you're seeing here now. Okay, so if you cut up through the body and pin away the outside surface of the Nereis, then you'll have exposed the intestine, which is obviously responsible for processing food. Okay, it's going to lead to the anus. Anteriorly, it leads to the esophagus and the pharynx. Okay, so the esophagus is right here. It's a thinner, smoother structure compared to the intestine. It's also thinner compared to the pharynx. The pharynx is more bulbous. You can see that here. Okay, and of course that would lead to the mouth. Okay, so let's look at our next slide to show you that the pharynx can be extended outside the body, everted, or drawn into the body for digestion. Okay, so this is a side view, of course, externally, of our Nereis, where you can see now these tentacles are kind of like flipped up in the air because the organism has extended its pharynx to allow these jaws, the sharp like teeth pincher structures, to grasp food. And then if you look closely, there's little tiny dots, little black dots here. These are called denticles, and they also help with mechanical digestion of food once the pharynx has been brought back into the body. Okay, so this is showing an everted pharynx, and this is showing the inverted pharynx. Now this is what the organism looks like with its normal tentacles, the palps, the prostomium, and the peristomium. So this is without the everted pharynx, and this is showing the everted pharynx, okay? You see those jaws extended, and then they bring the food into the body for digestion, okay? All right, now we have two slides that you are to look at this week. Because you're not in lab, I've made some videos of those for you, but here is a good picture of a cross section that looks very similar to the one slide. It's in your lab manual as well, very similar picture. the video should also help you. Just showing you the internal anatomy. If you were to cut the Nereis through the body, sort of towards the middle of the tail, cut it through that, and then look at the cut side, this is what you would see, okay, if it were stained and on a slide. Okay, so there's different muscle structures I'm going to talk about. There are digestive structures, the parapodia. You can obviously see, I'll start with those. So here, letter C is the parapodia, one of them. Here is the other parapodium. They help with locomotion, and they also help the Nereis with respiration. So the Nereis just absorbs oxygen through its body wall, and the increased surface area that the parapodia provide allow for increased respiration for the Nereis. So they serve sort of as gills for the Nereis. Those are the parapodia here and here. And then if you look at, this is the dorsal side, this is the ventral side. If you look at the internal cylinder, this is going to be the intestine, J, okay? The intestine, of course, is part of the digestive tract. The space that then is outside of the intestine, but inside of our body wall, is the coelom, G, okay? And then let's look at some muscles. So A here and here are our dorsal longitudinal muscles. So they would run like through the computer screen at you, okay? So they're running longitudinally along the long side of the Nereis, okay? Dorsal on the dorsal side, dorsal longitudinal muscles. Now we've got a complementary pair down here. Letter D, these are our ventral longitudinal muscles, okay? So again, running the length of the organism long ways, okay? Out the screen at you. All right, we've also got a circular muscle, B, okay, running around the outside of the Nereis, and oblique muscles, which are letter H. They run here and here, okay? Those are oblique muscles. So these are the muscles. We talked about the digestive tract. We did not talk about blood yet. So the letter I here is showing you the dorsal blood vessel. And then we have a ventral blood vessel, letter F, ventral blood vessel here. So these are the two major blood vessels of the Nereis. We don't see a real pumping organ. We don't see a heart in the Nereis. It's not that developed. However, the blood is completely encompassed in blood vessels. So what we say it has then is a closed circulatory system. All of the fluid is enclosed in blood vessels, which is not true in the crayfish, which we'll look at in just a minute. So the Nereis has a closed circulatory system. All the blood is in blood vessels, dorsal and ventral blood vessels, and a few vessels associated with the parapodia, which you do not have to be responsible for. And the way that the blood travels through these blood vessels is just through contractions of the muscles. So the circular muscles and then the longitudinal muscles, the movement of the body is really all that moves that blood through the blood vessels. There's no, like I said, there's no heart, okay? And then the only other structure that I think we didn't talk about is the ventral nerve cord, which is letter E here. The ventral nerve cord is the primary nervous structure of our Nereis, okay? So if you remember, we have a dorsal nerve cord. So ours is on the opposite side of the body from the Nereis. The Nereis has a ventral nerve cord, okay, running along its belly side. All right, so that's the slide that is representing the cross section of our Nereis. Now there's one other slide that we have you look at. It's showing you the parapodium of our Nereis. So the parapodia, again, are the little legs that come off of almost each of the body segments, almost each of the metameres. And they have a biramous structure, meaning there's two halves basically to them. One half and the other half. Okay. And the two halves are called the nodopodium and the neuropodium. The neuropodium is going to be closest to our nerve cord. So it's going to be on the ventral side. So it's going to be all of this stuff here. Okay. Nodopodium. Or neuropodium, excuse me. Neuro is on the ventral side. And then the nodopodium is going to be on the dorsal side. Okay. So all this stuff here. Okay. And there are two ligules associated with each of those. So like two halves of that. Here's one, here's the other. But this is all part of the nodopodium, and this is all part of the neuropodium. Okay. And then there are these chitinous rods right here called achicula that are support structures for each half of the parapodia. And then there are also little bristly structures associated with each half called setae. Okay. Different fibers that are produced from an individual cell, again, associated with the nodo and neuropodium. Okay. The whole thing is called the parapodia. All right, and again, as I mentioned, these obviously function with locomotion, but they also allow for increased respiration for our Nereis. Okay, so I think that wraps up our talk on the Nereis, on the worm. So let's move now to the crayfish. We're going to look at some classification of the crayfish and then some external anatomy as well as internal anatomy. There are no slides, of course, for the crayfish because it's too big. All right, so it's in the phylum arthropod, which means jointed foot. Okay, so each of the feet have joints and they have different sections to them as well. It's in the same subphylum as lobsters, shrimp, and pillbugs. It's the subphylum Crustacea. Okay, it's in the order Decapoda, which means 10-footed. And now instead of having identical repeating metameres, we have much more specialization going on. You're going to see a lot more differentiation in the appendages, as well as much more specialization in the development of the organs. Okay. Crayfish have 19 body segments, which each have a pair of appendages associated with them. And then I'll just show you the basic parts. You've got the head and the thorax, which combined make the cephalothorax right here. And then this tail is actually called the abdomen. Okay. And there's a hard structure called a carapace, which is a protective covering all over the body. Okay. So that's called the carapace. All right. And now we're looking at, that was the dorsal side. Okay. Of course, dorsal side. Now we're going to look at the ventral side. Okay. And when we flip it over and look at the ventral side, you can see the attachment points for a lot of the appendages. Okay. So this is basically showing you these large, the most obvious appendage, I would say, is the caliped. These are the graspers, the claws on our crayfish. And then there are a lot of appendages associated with the head and mouth, which we will look at. But first, I'm just going to point out that behind the caliped are 4 pair of walking legs. So these big legs coming off the side, there's 4 pairs. And then behind that, on the abdomen, there are 5 pair of swimmerets, sometimes called pleopods. Okay, so these are little legs, which you can see a little bit better here on the bottom right picture. Okay, and then behind that, you've got a bilateral uropod. That's another appendage with the central telson in the middle. The telson holds the anus. You can't see it real well, but right here is the anus. It's in the center of this telson, flanked by the uropods. Okay. And then these little structures here, here's like the attachment point of one and another, a third, a fourth, and then there's a fifth swimmeret or pleopod. Okay. Pairs. There's 5 pairs of those. Now, also mention while we're looking at the swimmerets, they look very different if you are looking at a male versus a female. So if you were given a crayfish and you turned it over and you looked at the belly side, the ventral side, you would see either 1, 2, 3, 4 large swim rats with a fifth one that's tiny up here. And if you saw that, you should know that you were looking at a female. Or if you had a male, you would see that the first 2 pair are much bigger than the last 3. Okay, so I'm going to take you to the next slide to show you that in more detail. Okay, so this is the male, and they have 1 and 2. You can't see that one too well. 1 pair and the second pair that are larger. The swimmerets or pleopods are much larger than the last 3, which would come off down here. I think they've been detached in this picture, but they're much smaller than the last 3 pair. And these 2 large ones in the male are called copulatory swimmerets. They allow for the male to grasp the female and release sperm that will assist with fertilization. Okay, so that's the male. It has 2 larger pair and 3 smaller pair of swimmerets. And then the female, we're going to look at our next slide, will have 4 large pair with 1 tiny pair. You can see it here. This one, it's kind of like wrapped around. This is the tiny little swimmeret, and this is a larger one right here. Okay, so here's one, and here's the other. And everything is present on both sides of the body, so it's a pair. Okay. All right, so in females, the first pair of swimmerets is smaller than the subsequent 4. And then if you look at number 2 here, labeled in our picture, this is the opening to the seminal receptacle where sperm is going to enter the female and allow for internal fertilization. And then once fertilization has occurred, then I'm going to show you the opening to the oviducts, which is more anterior, more rostral, more towards the head, between 2 pairs of walking legs. So these are the larger walking legs on the thorax. And so these little openings here are the openings to the oviducts where the eggs will then be released. And then if you look down here, once the eggs have been released from the body, they attach to the pleopods, the female's pleopods or swimmerets, until they have hatched. Okay, so there's just showing you all those little baby crayfish. Okay, all right, and there's just a note here just showing you that when you look at the crayfish, these openings will not appear so obvious or so large. They've been enlarged and expanded to show you them in more detail. Okay, so you really kind of do have to hunt for them when you're looking at the crayfish, but I just want to give you the best picture that we could for background. All right, so now we've talked about the calypeds, the claws, the walking legs, the swimmerets, the uropod, and the telson on the tail. So now we have to move towards the head and look at the appendages that are on the head and around the mouth. Okay, so there are 6 pair of mouth appendages that layer and cover the crayfish's actual mouth. And then on the head, there's also a pair of antennae and a pair of antennules. Okay, so those are, this is the ventral side. This picture showing you the ventral side of the crayfish on the head, of course. So I'll just point out the eye. That's going to be more on the dorsal side, so you only kind of see half of it. The antenna, one of them is here. The antennules are kind of hidden right now. But this is showing you where the mouth is, okay? Here is the calyped, okay, coming off. And so you kind of have to look just in front of the calyped to find the third maxilliped, and that's shown right here. I'm going to trace the cursor along it. This is the third maxilliped. It's the largest. Okay, so here's showing you them all compared to each other. It's going to be the most external of the mouth appendages. So if you are dissecting this, which we would normally have you do, you take that third maxilliped off first to uncover then the second maxilliped, which is kind of underneath it. So you pull off the third maxilliped. Underneath that is a smaller second maxilliped, Again, everything's bilateral. Everything's in pairs. Okay. So you've got your third maxilliped on both sides of the body. You've got your second maxilliped on both sides of the body. First maxilliped on both sides of the body. And now you can't even see the maxilla on this because they're so small and feathery and they're hidden by the other structures. Okay. So you remove those maxillipeds, and then you also have a second maxilla and a first maxilla, which would be directly then on top of the mandible, which is a much harder structure. It's directly on top of the mouth. So directly on top of the mouth, you've got that hard mandible, first maxilla, second maxilla, first maxilliped, second maxilliped, and third maxilliped is the externalmost mouth appendage. Okay, so you should be familiar with those and the sizes, respective sizes of them. And when we return to campus, hopefully you'll get a chance to look at those and see how small and feathery and kind of hard to tease out they actually are. Okay, I would like you to be able to see those. All right, so let's move to our next slide, where we're going to start to talk about internal anatomy. Okay, if you were in lab and you were doing a dissection, we would have you cut the carapace away to look at the internal structures, first on the dorsal side, and then we remove those internal structures to see further towards the ventral side of the crayfish. Okay. All right. So the first thing that you might notice when you remove the carapace is that a lot of that space in the thorax is taken up by digestive glands, okay? And they're a pale color, take up a good bit of space in there. They are responsible for aiding in digestion of food, as you might suppose. Okay, and then anterior to that, you would see the stomachs. And I say stomachs, plural, because there is a cardiac stomach and a pyloric stomach. The cardiac is most towards the head, most anterior, and it has the job of mechanically digesting food, okay? And then just behind that is the pyloric stomach, okay, where my cursor is now. That's responsible for chemical digestion of food. So using digestive enzymes produced from the digestive glands, then it further breaks down food, okay? And if you were able to, I usually have my students cut open the cardiac stomach, you'd see these little teeth inside. It's really cool. Little tiny teeth that basically help with mechanical digestion of food, and that's called the gastric mill. Okay. And then, of course, the pyloric stomach has digestive enzymes, and then the digestive glands, more rostral or more caudal to the stomachs. And then I'll take you just a little bit further back towards the tail, more caudally, to show you the heart, which is this little one structure on the midline. We often get crayfish that have been dyed so that you can see the heart and the blood vessels more clearly. This one has not, but you would notice the location of the heart is going to be on the midline. It has little pores in it, which allow for hemolymph to return to the heart. So these little pores are called ostia, and these are present because the crayfish has what's called an open circulatory system, meaning not all of the fluid is contained in blood vessels. There are some blood vessels. However, there's a point where the blood vessels stop and the fluid, the hemolymph, just kind of washes over the organs of the crayfish and then return to the heart through the ostia, through the pores, okay? So you should know that though there are some blood vessels, the crayfish has an open circulatory system, okay? And then I'll just mention, because I think you can make out the intestine, okay? So we're gonna start to look at the digestive structures as well. When I get to the next slide, I'll mention that you can see the esophagus. And so I'm just gonna walk you through the fact that after the mouth, food proceeds through the esophagus to the stomach. You kind of have to remove that stomach to be able to see the esophagus. And then, of course, after it passes through the stomach, it's going to proceed through the intestine and then out the anus, which is going to be on the ventral side in that telson, which we saw before. Okay, it's going to travel through the tube out through the anus. Okay. All right. So I think for this picture, the top picture, oh, I forgot to mention the gills. The gills are going to be very feathery on either side of the body. You can see some here, some here. And actually, if you were in lab and you were removing the walking legs, the appendages on the outside, sometimes you can actually pull out a gill with that because they're kind of attached on the inside. Okay, so these are the gills that you would find on either side of the body internally. Okay, and then if we look down at this picture here, there's one or two other structures that I want to mention. The green gland, which I'm going to mention in the next slide as well, is the organ responsible for the excretory functions of the crayfish. So waste products are processed and removed via the green gland. Okay. And then in the abdomen, there are a lot of abdominal muscles, abdominal flexor muscles. And then also you can see a little bit of it here. This white fiber running on the ventral side is the ventral nerve cord. So like the Nereis, we have a ventral nerve cord in the crayfish versus our dorsal nerve cord or spinal cord. Okay. All right, let's move to our next slide. So now what you're looking at, again, is the dorsal side from top down, dorsal side down. However, a lot of the dorsal internal anatomy has been removed. So the stomachs have been removed, the digestive glands have been taken out, a lot of the abdominal muscles have been removed for you to be able to see now the most ventral internal anatomy, okay? So one of the major structures, which I had mentioned already, is the green gland, okay? And that's present bilaterally. Here's one, here is the other. They're not green, and they are responsible for the excretory system for the crayfish. So waste products being removed. And then if you look, this white fiber running here and down through the tail, through the abdomen, this is again the ventral nerve cord. Okay, and we don't get too sophisticated of a brain with the crayfish yet, okay? But if you move towards the head and go around the esophagus, you'll see that there's some branching and then a ball of cells, basically, which is a ganglion. It's like a ganglion of cells, nervous cells, which basically make up the crayfish's brain. Okay, so they would be white. Most of it is a fiber, and then at one point towards the head, it forms kind of a small mass, which is the most extensive that we get for this animal. So that's its version of our brain. Okay. All right, and then I'll just mention, because I told you that the esophagus is then going to connect the mouth to the stomach. Okay, so that's going to be the tube that food passes through. It's a little hard to make out here, but it's right here. Okay. All right, I think we've covered most of the internal anatomy of the crayfish. So I've just included this table showing you a list of structures in the crayfish and their functions, but I believe I've talked about all of them. The heart pushes our hemolymph, which is their version of blood, through the arteries. The ostia are the pores that allow the hemolymph to re-enter the heart after having washed over some of the organs. There are two major arteries, which I did not mention, and run the length of the body: the dorsal artery and the ventral artery. Okay, so supplies the abdomen with the fluid, the hemolymph, and the ventral artery distributes the hemolymph to the walking legs and the swimmerets. So again, we're looking at the blood vessel on the dorsal side and the ventral side. Okay. Mandibular muscles help to move the mandibles and crush food. The cardiac and pyloric stomachs are responsible for mechanical and chemical digestion, respectively. The gastric mill, I had mentioned, is inside the cardiac stomach, and its little teeth in there that help to grind up the food. Okay. Digestive glands we saw, and those supply digestive enzymes to the pyloric stomach, which again is responsible for chemical digestion. The esophagus allows food to move from the mouth to the cardiac stomach, The intestine is responsible for absorption of nutrients from food and removal of waste through the anus. And then the green gland collects and excretes urine and metabolic wastes. Okay. All right, and then I have one more table that basically compares the few important details between our Nereis and our crayfish. They're both protostomes, but they have a few important differences, okay? Namely, their circulatory system. The Nereis has a closed circulatory system, meaning all of the blood is enclosed in blood vessels, whereas the crayfish, not so, has an open circulatory system. They both have bilateral symmetry, complete digestive systems. They use gills and parapodia for respiration. They both have male and female reproductive parts, for each. The excretory system for the crayfish, we have the green gland. For the Nereis, we have what's called metanephridium. That's their version of our kidney. And we cannot really see those very well on the Nereis. If you were in lab, we might have you try to look under the dissecting scope to find the metanephridium, but they're very hard to see. Almost, we never find them. So, nervous system, they both have a ventral nerve cord. Their brains are basically ganglia, compilations of cells, slightly more specialized in the crayfish. And then the locomotion, obviously for the crayfish, we've got walking legs and swimmerets to help them get around, and then parapodia in the Nereis. Okay. All right, so to sum up the stuff for this week's lab, we want you to be responsible for external and internal anatomy for both the Nereis and the crayfish. For the crayfish, we want you to be able to know the difference between the male and the female, how you can tell. Okay, so you want to look at the abdomen, look at the ventral side, look at those swimmerets, be able to tell whether the crayfish is a male or a female. You should be able to identify each of the appendages in the crayfish and the internal anatomy. We want you to also be able to look at the slides for the Nereis. So there's one that is the cross section and one of the parapodia for the Nereis. And then you have several assignments that are due next week already. You're going to submit these through Canvas. The first is the plagiarism assignment. You should not let that go till the night before. Okay, some students do require multiple tries to finish that. It's all online. There's a website that we send you to. Don't let it go till last minute. Okay, I often get emails the night before. I'm having trouble. Do it ahead of time. Okay. The citation ready assignment, again, there's a description of this assignment posted on Canvas that should be pretty thorough. If you have any questions, shoot me an email, cfry@lvc.edu, and then your first post-lab assignment, okay? All right, hopefully you guys are all enjoying your time at home, and I hope I get to see you soon. Any questions, please email, and have a great week.